Welcome to the next installment of the ATLT Insight series. I am Eme Gisore from All Things Labs and Testing, and I am thrilled to open today's session on the Masterclass webinar presented by Dr. Owen Fraser and Dr. Liberty Sibanda. So what is ATLT? Welcome to the next installment of the ATLT Insight series. I'm Eme Gisore from All Things Labs and Testing, and I'm thrilled to open today's session on the Masterclass webinar presented by Dr. Owen Fraser and Dr. Liberty Sibanda. To ensure that we stay on time, we will start immediately, and as people filter in, they can follow along. So firstly, I will introduce ATLT, and All Things Labs and Testing, ATLT for short, is a transformative platform dedicated to driving excellence in laboratory science, trade, accreditation, and testing technologies. Since our launch, our aim has been to unite industry leaders, regulators, innovators, practitioners, and academia to share best practices, showcase breakthrough technologies, and collectively address the challenges that hold our sectors back. We focus on critical sectors that form the backbone of Africa's sustainable development, including agriculture and food, water and irrigation, environment, engineering, mining and energy, forensics, and ICT. Every one of these sectors depends fundamentally on robust, reliable testing because what gets tested gets trusted. The ATLT Insight series is a knowledge-driven exchange engine connecting you directly with leading subject matter experts and thought leaders who are shaping the future of testing in Africa. Our series is anchored on four pillars. That is thought leadership, where we share knowledge and foster conversations on best practices, knowledge exchange, sharing proven solutions and best practices that work, industry challenges, tackling sector-specific barriers to growth and development, and lastly, innovation spotlights, highlighting emerging technologies and their transformative impact. Today's session will provide a thorough yet concise exploration of mycotoxin challenges in food safety, focusing on principles, methods, and best practices for accurate mycotoxin detection. For mycotoxins remain a persistent global challenge in food safety. So our key presenters today in this masterclass is Dr. Owen Fraser, who brings extensive expertise in laboratory technology and analytical standard development across Africa and internationally, and Dr. Liberty Sibanda, who offers deep knowledge in mycotoxin detection techniques and international residue monitoring programs. This masterclass will last for exactly an hour, in which it will be a 45-minute presentation, followed by a 10-minute live Q&A. So do prepare any questions that you will have, for they will directly address them. And thus, it is my genuine pleasure to welcome Dr. Dr. Owen Fraser and Dr. Liberty Sibanda to the virtual stage. And thus, over to you, Dr. Fraser.
Hi, Amy. No, this is
Okay.
Liberty.
Okay.
So Owen is actually in a situation where he can't join at the moment. So what I'm going to do, I'm going to cover the uh mycotoxin regulations, ones that he wanted to actually he had planned to cover. So if that's okay, I can actually go directly to my presentation if that's fine.
Yes, that's quite all right.
Yeah,
Thank you very much.
you're welcome. Let's see. Recording now. View. So I'm trying to see where I can share my screen on this. Let me say okay over here. Being recorded. Right. Hear that. Speaker. Hmm. So Amy, I'm trying to see where exactly. Oh yeah, okay, sorry.
Okay.
There we go. Okay, can you all see that screen?
Yes, yes, yes.
Okay,
You
brilliant.
can just put it in presentation mode.
Yeah, right. And you can put reduce it. And this one I need to reduce it as well. Okay. Right. So mycotoxins, qualitative, quantitative, and confirmatory methods. So I will start, by just listing the topics that I'm going to cover today. So, which is going to be mycotoxin regulations. So, looking at the EU, the US, so USDA, FDA set of regulations, as well as the Codex. And then I'll go forward to sample preparation approaches, different types, and then look at the methods themselves. So covering lateral flow, ELISA, microarray, HPLC, and LC-MSMS. However, just to give you a brief background on mycotoxin, resulting in the regulations as well as the testing approaches, looking at an example of maize, you see here maize being infected by Aspergillus, and then here you have Fusarium, and then again here you have both of them actually um, contaminating or infecting the maize. So each one of these, for example, the Aspergillus is known to produce more than one mycotoxin. So you would expect that it will have probably aflatoxins and ochratoxin in that particular infection. And then the Fusarium itself produces many mycotoxins like the fumonisin as well as the different trichothecins as well. But now looking at the regulations, the EU covers most of the mycotoxins we know or we worry about. The aflatoxin B1, the total, and then ochratoxin A, fumonisin, and DON, zearalenone, and tetrodotoxin. And you see the MRALs here that they are quite low. This is for human consumption, though. And then if you compare that to the US, you will see that there's quite a difference in terms of the MRLs. They are a lot higher than what you would find in the EU. Again, comparing that with Codex. So Codex affects, I mean, not really affects, but it's the regulatory regime that is adopted mainly in most countries, except. No, that except really is just that Codex is used as the foundation for all the regulations in terms of food safety. And then countries like the US and maybe regions like the EU will then further strengthen their own exposure levels, but really taking that from the Codex. So they'll look at their food basket and see how much they have to go down. So, but in most of the countries elsewhere, the Codex is the one that is used. And you see here for aflatoxin B1 is 10 compared to the EU of 2 ppb, and then 20 for aflatoxin total. And that is almost the same as what the US is prescribing. And then ochratoxin A, the US is pretty high, while the EU is really low. And we have Codex at 5 ppb. And fumonisin is 2 ppm, and that's the same for DON. And yet the US and the EU are lower. There is no, currently there's no regulation for zearalenone or T2 toxin, but the toxicity data, it is there though, but at the moment there's no implementation. And then moving over to wheat. So I'm just going to give you just a few matrices that are covered by regulations. And so wheat, we see here regulations by the EU, really low, almost the same as we saw in corn. The reason why there is no regulation here for fumonisin is because primarily fumonisin is produced in maize and not wheat. So perhaps even if it might be produced to some extent, it is very, very low to you know, elicit any worries in terms of exposure, levels. And so we see also Dawn, as well as zearalenone and T2. So if you compare that to the US, you see here the US is only considering total aflatoxin, and we have quite a lot missing. Of course, there's a lot of like ochratoxin A produced in wheat, dawn, zearalenone as well, and T2. So for me, it's mind-boggling, that there are no other regulations here. The US is not enforcing that. Um, and then we go over to the Codex. Codex, we see ochratoxin A and DON being regulated in wheat. So we miss a lot more here. So moving over to things like nuts. So in nuts are mainly infected by Aspergillus, okay? So that will really limit the number or the types of mycotoxins you will find being of concern within the nut matrices. And so the EU considers aflatoxins and ochratoxin A. And simply because Aspergillus, that's what it primarily produces in nuts. If we look at the US, they're only considering total aflatoxins. And Codex considers both aflatoxins and ochratoxin A, just like the EU does. Now, the biggest concern really, because we can control within single matrices and maybe limited combination of different types of foods, and then for human consumption, that can be easily regulated and controlled. But when it comes to feed, so animal feed is a little bit more complex in that one, the quality of raw materials that are used, they are of lower standard or lower quality. And that in itself already tells you that the risk of high mycotoxin presence or occurrence is really high. And at the same time, you mix different types of agricultural crops. So it really becomes a bigger problem, a more complex problem, which requires a very um, calculated sort of approach. So in terms of regulations, we see here, um, the EU is regulating the aflatoxins, ochratoxin, fumarisin, DON, so all of them. Um, and of course, the tolerance limit, yeah, for animals is much higher than it is for humans. So you see here that the maximum limits are, of course, more um, a little bit higher than for human. And then in the US, they're only regulating three mycotoxins officially, the aflatoxins, fumonisin, and DON. So usually the US guys do not talk about DON, they talk about vomitoxin, simply because of the effects of DON in animals, so causing vomiting and all that, and feed refusal. And when we look at what Codex does, only total aflatoxin. Okay. So if we now look at the methods that are used to control and enforce regulations, industry is not really enforcing regulations, but they are mainly controlling in order to put products on the market that are that are fit for consumption. And then, of course, you have government laboratories and some private laboratories to whom samples are sent for enforcement, regulatory enforcement. So here, one of the methods, of course, is the immunoassay. So we are all familiar with immunoassays, but the main ones that are used are well, before I actually explain the different types, but for immunoassays, sample preparation, if you look at sample preparation first, it is quite easy. My apologies. It is quite easy. And all you take is maybe 5 to 25 grams of homogenized ground sample. And this is extracted by shaking. Either you use water or you use methanol water. In some instances, it is ethanol water, 70% ethanol. And then in some instances, it is acetonitrile methanol water. So it depends what sort of extraction method has been optimized for that particular testing method. And then the sample is centrifuged or filtered after shaking. And then the supernatant is diluted by either water or wash buffer. So in most cases, wash buffer is used because sometimes when you dilute with water, you have a lot of precipitation or you know the carbohydrates or some of those organic molecules they come out of solution because they've been using an organic solvent, so to speak. So one of the methods that is used is lateral flow dipstick. So this is one of the immunoassays that is used. So you are pretty much familiar with lateral flow testing, like you see for example, the COVID test or the pregnancy test, something like that. So here, an antibody is used, and then you have a conjugated antibody as well, and this is conjugated to a signal, or at least for signal generation. So it could be conjugated to carbon or something like that. And then, so this is the layout of a lateral flow test. You see here, we have the conjugate pad. So this is where this conjugate is placed. And you have, so on the test lining itself, you actually might have the target analyte. And the control line, you have an antibody that is um, anti-analyte antibody, so that it just gives you a highlight of the success of that particular test. And then this is the absorbent pad that absorbs the chromatographic migration here so that you keep it going until the sample is completely drained through laterally on the surface of the lateral flow test. So when you apply, say, a negative test, you are expecting to find or to get at least two lines, for example. And then if you apply something that is positive, then in fact, you will have only one line. So this is supposed to be the other way around, actually. Okay, so here, normally nothing will appear for mycotoxins if it is positive, and if it is negative, you have two lines. So, for example, like we have here, this is the control line here. And then you will have the test line that will show you that this particular sample here is negative. And the next one, of course, is positive. It's only got one line. And then the third one here is invalid. So actually, this is invalid. And it still has negative there. So it's invalid because you don't have the control line over here. We're supposed to have a control line. So that is actually visualized by eye. So this is one technique of interpreting the results. And then the second one is where you actually use a reader to do quantitative determination of the concentration of the mycotoxin in that particular sample. Okay, and so this is the line here, or the area where, you know, at least a sort of approximately, accurate, an approximate concentration can be calculated over here, especially when you're using a reader. So the performance of lateral flows, in terms of the time they take to test, is very fast. For aflatoxin, for example, this particular one is 6 minutes. So most of them really vary from 3 minutes to 6 minutes. And um, their LODs, they are around this much. So you can see here for fumonisin and for DON, they're around 300 ppb. And also because the MRALs for DON, they are much higher. So you know that it's about 1 ppm for food and much higher for feed. And then, but for aflatoxins, it's 2 ppb. So the regulation is very low. It's around 2 ppb. So this might not be adequate, because it is on the um, line of regulation itself. The ochratoxin A is okay. However, some users here, because the regulation for ochratoxin A is around 2 ppb. So this might generate false positives, simply because it's too sensitive. So you have to figure out how is your test performing around the regulatory limit and for which sort of matrices. So you have to do a mini validation. So same goes for the rest of these, depending on where the regulation is, because you want to say, is my sample positive or is it negative? And how much variance do I have for my positive samples or negative samples around the MRL vis-a-vis the LOD of my test. So if you're using quantitative determination, you might use a reader. So different types of readers, or some of these tests come, or are supported by certain readers. So I'm just showing two examples here of some readers that are available on the market. So, and then we have the enzyme-linked immunosorbent assay, the ELISA. So this is the one of the traditional, of course, tests. So let me have a look. So these presentations. So what I'll do is Take a second. Sure. Can I go? So basically, like I said about the lateral flow, I just saw a comment just now. So the lateral flow, it was on the other way around. Um, so the negative is actually positive, and the positive is negative. So for ELISA, so we have here um, the mycotoxin enzyme conjugate, denoted by that symbol, and then we have the mycotoxin itself, and then the substrate and the antibody. So ELISAs come with microwells, and you might have the anti-mycotoxin antibodies spotted in the microwells. And in that particular case, um, right, in that particular case, you have the antibodies there, and then um, the mycotoxin conjugated or conjugated to HRP will compete with the mycotoxin in the sample for attachment sites on the antibody itself. So this involves, you know, incubation time, and also involves or requires a little bit more accuracy in terms of volumes that are used. So here you use micropipettes, and the pipetting has to be quite accurate as well. And so you incubate that, and then you decant whatever is not reacted, and wash the wells, and then you add in your TMB substrate, and then stop the reaction, which turns the solution to yellow. So you stop the reaction using a weak acid, and then turns to yellow. It is the yellow that then the yellow color that is detected by um, or measured by the detector. So this is a mere spectrophotometer that is used to measure the color. So um, ELISAs come in that format. So in a kit format with a microtiter plate. Normally this is 96 wells, or some products may come with 48 wells, which is just half the plate. You have your wash buffer or concentrated wash buffer, you have your assay buffer, you have your TMB as well, and then also have the enzyme conjugate and so on. So normally what you do for extraction, like I've already alluded to before, so you weigh your sample, yeah, you shake it with the extraction solution, centrifuge, and then for your assay, then you add the standards and samples plus conjugate together in the well. In some instances, the standards and samples are added first in their respective wells and incubated, and then the enzyme conjugate is added later. But to make the assays faster, so standards and samples are added, together with the enzyme conjugate and incubated together. So this kind of shortens or drastically shortens the assay. And so incubate that, and then after the incubation, you wash, you add the substrate, so your TMB, and incubate that. It turns blue because the TMB is being acted on by the HRP, and then you stop the reaction using a weak acid, and then this is then read in the reader, ELISA reader, and then you can construct, uh, depending on the configuration and the programming of the reader, it can already give you a calibration curve and estimated concentrations of your unknown samples. Okay, so the thing about the ELISA and the lateral flow test is that one kit is dedicated to one mycotoxin. For example, aflatoxin comes with its own kit, or there's one kit for aflatoxin, and then there's another kit for ochratoxin, for DON, for fumonisin, and for the rest of them. So you have to test if you want to test, say, 6 mycotoxins or 7 mycotoxins in a sample, you have to have the different kits, each one for each of the mycotoxins you're testing for. So in terms of performance, so there are some fast kits. For example, aflatoxin B1 fast kit, taking 5 minutes is absolutely, it's mind-boggling that a ELISA can take 5 minutes. But the ones that I'm most familiar with, they will take 10 minutes, maybe up to 30 minutes. But of course, the usual time period that we know about, fast mycotoxins or normal mycotoxins, I mean, ELISAs, they will take maybe 45 minutes to an hour and a half, like you see these. So you will notice also that shorter incubation times will result in not so sensitive um, detection limits. So, but if you incubate longer your samples, then you will get a much more sensitive sort of assay, and so that's what you see here. Okay, so still with the immunoassays, there's what you call the biochip array technology. This is from our own company for simultaneous detection of up to 9 mycotoxins in a single sample. So on this one, it's a ceramic tile on which anti-mycotoxin antibodies are spotted on that tile. It's just 9 millimeters by 9 millimeters, almost 1 centimeter squared. So you can test many analytes actually, but for mycotoxins, we can test up to nine mycotoxins. It is a competitive immunoassay and with a chemiluminescence signal generation. So you have air biochip like that, and on that biochip you have a reference spot, correction spot, and another correction spot just for internal control purposes, and then the rest of the spots here are for the different mycotoxins. So a system looks like that. You have the imaging module. It is computer controlled, and also for shaking the uh, the samples in the wells. So sample preparation is very, very simple. 5 grams of homogenized sample, 25 mils of solvent, and then you vortex and shake for 10 minutes, and then centrifuge for 2 minutes. Dilute 50 microliters to 200 microliters, and then your sample is ready. And then you add your standards and samples to the standards and sample wells, incubate at 25 degrees for 30 minutes, and then add your conjugate. So it's almost like an ELISA, the procedure that you have for an ELISA. Conjugate, incubate, conjugate for 1 hour, and then you wash the biochips and then develop the chemiluminescence result with luminol and peroxide. And then you can read the results in the imaging module, and the system will generate all the mycotoxin standards or calibration curves automatically and simultaneously. And here, the minimum curve fit is 95%, and the usual or average regression that you get is around 99%. And so when you go to the results, it will list all the samples that you ran, and then you select whatever sample you want to have a look at, and then it will list all the mycotoxins that were tested in that sample, and then it will give you the concentrations in ppb for each of the mycotoxins in that sample. So the results can be printed off like this. You can see T2 toxin, 15 ppb. You see fumonisin, 221, ochratoxin A, aflatoxin B1, zearalenone. So they are all printed together on one sheet like that. And performance of this system is really good. You can see Most all the samples, whenever we participate in FAR-pass proficiency testing, we are always within the Z-score range. And so the error bars here represent the -2 and +2 Z-score ranges. And you can see our result is blue, and the FAR-pass one is the red one. There's another one, the UK Green Technology Network proficiency testing is the yellow one. So we are always, passing, so which is really, really important. And then we recently achieved performance tested AOAC certification. So our system here, Myco 7, it's been AOAC certified. So the biochip you can do with a single extraction step, you can detect all mycotoxins at the same time, multiple mycotoxins from a single sample, and we can detect very low sensitivity. Like, for example, the aflatoxins will be 0.25 ppb, as well as the ochratoxin A. So that is just as low as it goes. And yeah, at very low concentrations. And you can reduce the number of samples requiring confirmatory analysis in doing this. So it's a quite flexible sort of system. So looking at chromatography, it does involve weighing the sample, adding extraction solution and shaking, and centrifuge and taking the supernatant. So I think this is a very uh, common sort of practice. So having covered that, because you have to use for extraction here, in most cases in chromatography, we use organic solvents like 80% methanol or 70% methanol, maybe sometimes with a combination of acetonitrile as well. So depending. So you have your HPLC over here. And with the HPLC, of course, you need your mobile phase. Everything is computer controlled, and you have your auto sampler as well as your column oven. You have your pumps to pump everything through. It will come through the detector. So there are different types of detectors that are used. Either you're using a UV detector or you're using a fluorescent one, but a lot of people prefer to use the fluorescence detector also because you have better detection and better sensitivity. So when it comes to sample preparation for chromatography, um, let me have a look. I might be going ahead of myself. Yeah, I might be going ahead of myself. Let's see. So you take 25 grams of sample and you extract that, for example, with 50 mils of 80% methanol water and shake for 30 to 60 minutes, depending on the method. So and the mixture is centrifuged or filtered through Whatman number 4 filter, and the supernatant or the filtered and is diluted to less than 30%, um, especially with sodium hydrogen carbonate. Well, I remember using sodium hydrogen carbonate um, to dilute my sample, because if you use water, then it becomes cloudy. If you try to um, run that sample through, for example, an amino affinity column, the amino affinity column is going to block because your sample is now, it's got solids basically. So you extract with the organic solvent and then you dilute with water, and then things come out of solution, and then we have more solids. Again, it becomes cloudy instead of clear. So here, when you're diluting the sample, after your supernatant or your filtrate, try to use salt solution, and then it will keep everything in solution so that when you bring it over onto your amino affinity column, they will run, at a reasonable rate, and within a short period of time, you should be able to have cleared or having ran through all your diluted sample through the immunoaffinity columns. So another method, of course, is the quick, easy, and cheap method, which the catches, um, and also molecularly imprinted polymers. I haven't seen a lot of molecularly imprinted polymers being used routinely, but this technology, I've seen it coming on the scientific scene, I think like 6, 7 years ago, but I really haven't seen them. I don't know if anyone here has had an opportunity to try out molecularly imprinted polymers. So for chromatography, so solvent extraction, so most mycotoxins are of course, are polar, and either they have a polar reactive group or they have a polar charge. And so that actually allows them to dissolve easily. In most organic polar organic solvents like your methanol, acetonitrile, um, and also in water. So this is why you will find that there are some methods, especially for lateral flow tests, that they use water testing. So the water extraction. So water extraction, I haven't really seen the data of it in terms of extraction efficiency, but um, I think it is adequate, especially for rapid testing on the lateral flow. I think it is adequate, but when you want to do very correct, high recovery on things like ELISAs and maybe the biochip or the HPLC and the rest of the chromatographic um, sort of tests, I think there are some limitations. I really do not have any example or have experienced anyone that use water. So, but there are methods, especially lateral flows, that use water as extraction. And so cleanup is quite important, especially the solid phase cleanups. Especially we have immunoaffinity column. There are some columns that there is a column that can do 6 mycotoxins. It's quite a thick one. So depending on the method that you're using. So if you can extract 6 mycotoxins in the sample at one go, that's really good. You can use that on the HPLC, or and then you can inject it, for each method. Like for aflatoxin, I'll just take the same and inject it, and then when I'm running the ochratoxin A method, I will still inject the same sample. But I think the multi-mycotoxin column will work best for targeted LC-MSMS testing. And then, of course, you have your catches, which is uh, catches. Well, it is non-targeted. Here we are just extracting, a lot of your analytes. So, and then you also have the molecularly imprinted polymers as well. So your immunoaffinity columns are the ones that I know that I use mostly, like your AFLA test or your aflatoxin B1 column, your DON column, ochratoxin A, T2, fimonisins, zearalenone. So the immunoaffinity columns, um, they will be used in the case of HPLC, because for HPLC, um, let me have a look. Okay, yeah, which detecting method, right? Okay, wait. So, and so these ones will be for specific method on the HPLC, right? So, for ELISA, for lateral flow, and for the biochip array technology, the Randox biochip array technology, these are not required. So these will be required, of course, for the HPLC. And then you have the catchers. Where have you gone, catches? Ah, come on. Yeah, there you go. So the Ketch's method, so it's a powder in most cases, which then you mix with your sample in the tube here, you shake, and then you take your purified sort of liquid, which contains we're supposed to have the sample still in suspension. I mean, the analyte still in suspension, and use that. The molecularly printed polymers made in the presence of the analyte that they are supposed to catch. So the polymer sort of forms itself around the molecule, the shape of the molecule, and then so this then can be used in a column form. The powder of this can then be used in a column form where if you put that analyte through there, it will like lodge into the crevices that very, very specific to that analyte. And then you can elute it and so on and so on. So, but the methods that I know that are really in use are these, like the immunoaffinity columns and the catchers and other silica bound um, uh, silica bound solid phase methods. So when it comes to detection, so HPLC is a single analyte method in the sense that, for example, these methods here, I mean, the ones that I've set out here are all fluorescence detection, right? So for zearalenone, it's got a signal. It requires excitation at 274 nanometers. And then when you excited that, it will emit at 446. So this is what this fluorescence detector does. So, and then it's the same with the ochratoxin A, you excited at 330 and 33, and then let me see. And then it emits at 460. So you have to specify these excitation and emission wavelengths for correct and optimum detection of each of these mycotoxins. And as you can see, none of these mycotoxins have the same excitation and emission wavelengths. So they cannot be detected together. Okay. They might have different retention times. So that means they come out at different times from the column. But when it comes to detection, the excitations wavelengths and emission wavelengths are totally different. So you can't mix the two or the different mycotoxins. So it's always for HPLC, it's always one mycotoxin or one analyte that can be run, say for, I mean, I don't know how many hours it takes for an HPLC method to run. So it can basically be half a day or the whole day just for one method. Okay. So, and you do that, so that, the cleanup is really necessary so that you have a very stable baseline here. And then the integration events that are done then at the basis of the peaks then can offer or generate a very accurate peak area to give you the correct concentrations for that specific analyte. Okay. And you can then calculate most efficiently. So these are some of the performance data, depending on the method, of course, but I think immunoaffinity column usage does help to eliminate a lot of noise, and sensitivities can really go down as well because of pre-concentration. Pre-concentration as well is used when you're using solid phases as well, because organic phase, and then you dry it down um, and then reconstitute it in a smaller volume, and then can allow you to detect at very low concentrations. So the next one, of course, is the LC-MSMS. And as you can see, for LC-MSMS, you're still using HPLC, the front end here. But instead of using a UV detector or a fluorescence detector, instead, you are using a mass detector. Okay. So for the LCMSMS, it's almost the same as what you would have um, for HPLC in terms of sample preparation. But when it comes to the standard, so you have to create a matrix matched calibration here. So you need also to have all your mycotoxins in the matrix. So that the generation really mimics the environment in which the generation of the calibration curves mimics um, the condition of the sample. And then in addition to that, you also need to add an internal standard. Usually the internal standard, or not usually, the internal standard is always um, a non it's a non-radioactive um, isomer, or not isomer. What do you call it again? Um, it's a heavier, I will put it like that. It's a heavier molecule of one of your standards. For example, let me say carbon 13 instead of is it carbon 13? No, carbon 12. And then you use maybe carbon 13 or carbon 14. So just to make it heavier so that it is less um, or it does not interfere with the results of your the mycotoxin that you're testing for. So this is just to show you, or it's just for checking the recovery and the accurate determination of the rest of the mycotoxins. So I spoke about the six mycotoxin immunoaffinity column, and we have it here for myco 6. So this is for targeted detection. So I'm looking for 6 mycotoxins, and I'm going to use the 6 myco 6 or 6 and 1 immunoaffinity column. But then at the same time, um, it is the same sort of method that you would use for the single mycotoxin immunoaffinity columns. You elute, you evaporate, you resuspend, and you analyze. So using cleanup beforehand for LCMSMS is very, very important because you will not make your sauce dirty, because the sauce becomes dirty, then ionization efficiency really goes down. So if you're using a dilute and shoot sort of method without cleanup, and then pumping it in here, it is going to get dirty, as you continue to feed the system with the samples. Okay. And then, so for untargeted, usually you would just weigh your sample, do extraction, and then use cultures, for example, and other solid phase approaches, but not immunoaffinity column because it is very specific. And then of course, you centrifuge, and then you take your supernatant and um, inject that. So for LCMSMS, you need to have specific conditions that are set. So as much as it is expensive, it also requires quite a lot of detail. And it is, for example, with LC conditions, okay, you have specific columns and then you have specific buffers as well. And then, of course, you have to specify the injection solvents and the injection volume. But then you also have to, show exactly, what are the parameters for environmental ionization here. Is it positive, which positive and negatives are you looking for? And then you have to specify the collision gas and drying gas and so on. So it's quite a lot, that is required. So with LCMSMS, you should have the proper analysts that are going to operate it, as well as the maintenance that is required on the system. So in most cases, I mean, I've come across this the world over, whether in America, South America, in Asia, in Europe. It's always a problem to have a good budget, to get an LCMSMS, or to support an LCMSMS. It really is capital intensive. So it requires a mobile phase, and of course, um, you need methanol in there for baseline to support baseline separation as well. And then, of course, your acid intensifies the signal. So it's just there for promoting ionization in the source. So your stationary phase, okay, any sort of C18 bonded phase, for retention of your polar and apolar molecules. Um, and then, of course, the ionization is really important because this is what MSMS is all about in terms of detection, because it breaks the molecules. So there are certain parts where the molecule is vulnerable, for breakage, and then some of these methods are there already. Don't have to reinvent the wheel. And the ionization energies are there, for first of all to ionize it, and then there is the collision gas that is actually meant then to sort of break your molecules, and then yeah, you end up with the daughter fragments, and that fragmentation pattern is then determines the identity of your molecule. So I've come across people saying, "Ah, yeah, no, your test is not, it's not a confirmatory test, so we can, not use it." And I find a lot of people like, you know, simply because they were told that by somebody, or they did not quite understand what confirmatory, when does a confirmatory method come in. You can actually use an ELISA, or you can use HPLC, or you can use the biochip system from Randox, to do your normal testing. And then when you get a positive, then you put on the LCMSMS. Why? You're just doing that for confirmation of the identity of your molecule, as well as the concentration, of course. But if you know that you're using a validated system, and it is accurate, then you shouldn't be scratching your head, trying to get a budget for an LCMSMS, for example, if it's really out of your reach. But we have tools like ELISA or biochip, that can make a lot more sense. And then if you then have positives, you can send it to a lab. And positive samples, the rate of positive samples probably runs from a run, it runs from 0 to say 30%. Sometimes you can get 0, sometimes maybe 30% of your number of samples that you're testing becomes positive, but it is very, very low rate. Okay. Again, so like I was saying, so the source is for ionization. So you have to set out those ionization parameters. It comes with the curtain gas as well, and the dissociation. So there is quite a lot of stuff involved with LCMSMS. So I'm just putting all these things in there so that you just get an idea, you know, apart from the MSMS being able to detect mycotoxins, but what are sort of the structures that it actually has? So, for example, I was talking about dissociation. So, you will get like, some breakups, and then at the end of the day, these um, the target ions are the ones that will come up in terms of their um, intensities on the chromatogram. Okay, so these are some of the conditions. For example, I was talking about the ionization. So what sort of ionization is it? So some of them have negative, some of them positive, and then and all sorts of like you're looking at the dwell time, for example, how long does it take? Or is it set to take, for each collision in in the different sectors, either in Q1, Q2, of the MSMS. So the performance of the LCMSMS is also is actually very, very good, but it depends on the method. So we can say I have an LCMSMS here, but it actually depends on um, who has developed that method or who developed that method and how was it validated? How is it used? So the LCMSMS itself is a good method, but it just depends on the method that was developed on it and how it is being used. So it is quite costly as well. The chromatography methods are quite costly because of sample preparation. You have to do a lot of cleanup and it takes time. You do all that, cleaning up and, taking time simply because you need to achieve a very stable baseline. So that is the target, of course, to have that, for correct quantification of the unknown mycotoxins. Okay. So basically um, that is me. Okay. I don't know if you have any questions.
Thank you very much, Dr. Sibanda. There are actually numerous questions that have filtered in. I don't know
Right.
if you can access the chart, but if not, I can definitely read them out to you.
Okay.
So for instance, one particular uh, gentleman has asked that regulations tend to focus on the big six with
Mm-hmm.
climate change. And so, with climate change altering fungal patterns, are
Yeah.
there emerging mycotoxins that African and global monitoring programs should start prioritizing?
So the last part of that, what was the last part of the question?
He's asking if there are any emerging mycotoxins that
Okay.
African and global monitoring programs should start prioritizing.
Right. So when you look at emerging mycotoxins, for example, okay, maybe I should start with the effect of global warming. So we now could begin to see the emergence of higher concentrations of things like aflatoxins, for example, in the Western world. Um, so aflatoxins was a big problem in the um, developing world, like, South America, the African region, Asian region, but simply because of the tropical sort of temperatures. But now, because of global warming, there is a migration of the occurrence of things like aflatoxin, ochratoxin, in the Western world, where it is predominantly cooler, supposed to be cooler, but because of that, different regions are experiencing different effects of global warming. That is one. The second thing about emerging mycotoxins is that of course, there are emerging mycotoxins. Things like alternariol. You have um, um, what is it? Pivoricin. You have let me have a look. Uh, Altotoxin. So most of these mycotoxins I'm mentioning now are mainly a problem for the animal. animal nutrition industry because they are very wary of mycotoxins affecting the health of the animals and the performance of the animals. So therefore, they look for all sorts of mycotoxins. So you have tenoazonic acid, and then piperacin, eniatins, um, and then, of course, I did mention alternariol, tentoxin. So those are the main ones that maybe there could be some, but those are the main ones that the animal industry are worried about. And they are busy testing for them. Okay, so the animal nutrition industry is testing for at least 14 mycotoxins in their feed. So if you see companies like Cargill, Chow Nutrition, ADM, wherever, across the world, um, so they are working towards, having a control on all those mycotoxins, in addition to the 6 or 7 mycotoxins that are normally regulated. Okay, so I think it's now time that that the regulatory bodies should consider, what are the toxic effects in humans and what are our exposure levels so that we can have those regulations put in place.
Okay, thank you very much. Um, I think we can take in one more question due to time, and then since you've shared your contact details, people can reach out to you for more in-depth conversation.
Mhm.
So lastly, we'll use the next question where somebody is asking regarding rapid testing methods used in the field, how do we effectively bridge the data gap between these qualitative field results and the quantitative requirements of a reference laboratory?
Yeah, so that is an interesting question. Um, so in terms of so we need to be more accurate with the portable sort of tests or field tests. They have to be more accurate. So that's one. And also, we should be able to test within a very short space of time so that, field tests are supposed to be sort of one step or two steps, to get a lot of data, at once. So I think that should be the next step, um, in terms of, trying to get the same amount of data in the field that can sort of match or add to what we have in the laboratories. And again, being able to connect those field tests to the cloud, you know, and so that, or to a data center, so which would be probably in the cloud. And then being able to contribute immediately when you make the reading, then you're able to actually send that to the cloud in order, for all the data to be integrated within, say, maybe a supply chain sort of environment where we need to know how can we use raw materials from this region or not, and that has to come in a very timely fashion. So if that data can be up, immediately transferred or automatically transferred to the cloud, and it becomes available to decision makers regarding sourcing of um, feed or food, raw materials, that would be great.
Thank you very much, Dr. Sibanda. Just to stick towards the time stipulations that we give, we sadly have to put everything at a close at the moment. But
Mhm.
as he has shared his contact details, by all means, reach out to him? Okay, I hope so. You're comfortable with being reached out to in regards to any questions that people have because they're still filtering in through, and I think it
Hmm.
shows much interest. So, on
Yeah.
behalf of everyone, I would like to extend my deepest gratitude towards you for sharing your invaluable insights into the complex world of mycotoxin identification and quantification, and for also taking on Dr. Fraser's section to ensure the masterclass is a success.
Okay.
So
Oh, you're welcome.
we're definitely looking forward to even continuing on this conversation, maybe even having a longer and deeper deep dive. That way, everybody can even address all the things that they would like to know.
Okay.
So, lastly, I would like to thank all the participants for joining in this vital discussion and for igniting a very lively discussion. We would have to put everything here at a close. This is
Okay.
our last webinar for the year, and thus we're really grateful towards you, Dr. Sibanda, for ending it at such a high note. And we look forward to an insightful deep dive come 2026. So thank
Okay.
you all very much.
Amy, so if there are any questions that I didn't ask, you can send them to me and I can provide you with the answers and you can provide them to the individuals. But again, if anyone wants this presentation, I can send it to them. So if you can send me an email, so you have my contact details there. So you can send me an email, I can send you the presentation, okay?
Okay, yes. By all means, I'll share them with you.
Okay.
And thus, thank you all. I don't know if you have any last remarks before we end.
I think for me,
Doctors.
I'm good.
Mm, okay, lovely. So this is where we close. Thank you and have a lovely day to all of you.
Thank you.